Hello. So today I'm going to talk about using deep learning to optimize the method of DNA mini barcoding for tuna identification in the mitochondria regions. The goal of this video is to incorporate mini barcoding with an AI. It is a study from one of our professor, Dr. Halbert, and her study is on the identification of tuna species in raw and processed product using mini DNA barcoding of the mitochondria regions. The result of the study said 16% of the items were mislabeled, and the study success rate of using mini barcoding to identify tuna is 86%. What is mini barcoding? It's a simpler DNA analysis method, especially for identification species. However, her study faced some challenges, which is the cross-breeding species is a gene from different species mixed together. By this means that DNA traits that are unique to one species can get into gene of different species. For example, in this case, Pacific bluefin and yellowfin tuna, when they mate together, they can give a crossbreeding species. So when DNA barcoding try to detect that, it only give one result, whether it is going to be Pacific bluefin or yellowfin tuna. Compared to one another, they have different quality of flesh. Otolo is another name of bluefin. It's a high premium flesh of tuna that we encounter in a sushi restaurant. Compared to yellowfin tuna, most of the yellowfin tuna can be found in the canned product. And in canned tuna, they usually contain a degraded DNA sequences. It's a broken down DNA into a smaller piece, and it make it harder for DNA barcoding to detect the two species identity of the product inside the can. Now, I want to introduce artificial intelligence that will be incorporated in the study. The artificial intelligence is just a machine that can think like human, making a decision, problem solving. Deep learning is a part of machine learning that learn in a specific pattern and make a decision. And a convolutional neural network is like an algorithm of deep learning to recognize the pattern in image, shape, color, and number. So how can we incorporate AI to identify the true species of the tuna? We will use GenBank as a guideline to determine a species of the tuna. GenBank is just a collection of the nucleotide bases. After that, after they collect the DNA sequence, they encode them or give them a specific label like color, or it can be a number as well. Just add A is blue, green is G, T is yellow, C is red. So basically, the letter and the color is represent a nucleotide basis that found in the DNA. After hard encoding, the machine used these unique color pattern to learn and make a decision. After that, a machine will act like a magnifier lens to find a special clues or a pattern that tell which species is which, whether how complex the DNA is. And then after that, after they found a specific pattern, now we feed them to convolution neural network and let them make a match and make a decision of which species it is, and now we can determine if this tuna is a cross-breeding species or not. By combining AI with DNA mini barcoding, the success rate can increase to 97%, and this approach enhance accuracy and save a lot of times, and it would optimize the method of her study as well. Thank you for watching, and here is the reference.